All right, so I think we can start. Hello, everyone. I'm Mithun Mitra from the Indian Institute of Technology, Bombay. Welcome to this inaugural lecture of the monthly colloquium series on Quantitative Life Sciences. This is being organized jointly by the Abdus Salam International Center for Theoretical Physics, Trieste, and the Indian Institute of Technology, Bombay. And these colloquia will be held on the second Wednesday of every month, starting with today's talk. So in this colloquium series, we hope to hear from eminent researchers all over the world about recent advances in decoding the fundamental physical principles that govern the dynamic self-organization of living matter, from single cells to single organisms to collections of organisms. Over the past couple of decades, there have been spectacular advances in quantitative experimental techniques that have led to the realization that theory and modeling plays a critical role in biology, just as they do in the traditional disciplines like physics. So by tying together experiments with theory, quantitative biology sort of tries to uncover a deeper understanding of the mechanisms that influence the phenomena of life itself. And we hope that the colloquiums in these series will convey this broad sense of what fascinates physicists about living systems and the principles that govern them. I would now like to invite our co-organizer, Professor Antonio Celani from ICTP Trieste, to please say a few words. Antonio.
Thank you, Mithun. Thank you all for participating in the first event in this series of colloquia. I just want to express on behalf of ICTP that we are very, very happy that this series of events is actually taking place, and we are eagerly looking forward to further initiatives that can strengthen the ties between ICTP and IIT Bombay. So with that, I will give you back the floor, and looking forward to hear from Edgar.
Thanks, Antonio. So the first speaker of the series is Professor Edgar Roldan from ICTP. Edgar did his PhD from the Complutense University of Madrid. He was then a postdoctoral fellow at the Material Sciences Institute in Madrid, and subsequently at the Institute of Photonic Sciences in Barcelona. In 2014, he moved to the Max Planck Institute for the Physics of Complex Systems in Dresden, first as a guest scientist, and then in 2016, he became project leader there. So since 2018, Edgar has been at ICTP Italy. His research interests lie broadly in the application of this field of stochastic thermodynamics to understanding biological systems. So he's been awarded various prizes, notably among them the Early Career Prize with the Statistical and Nonlinear Physics Division of the European Physical Society in 2017. So today, Edgar will speak on the stochastic thermodynamics of active fluctuations in the ear of the bullfrog. So before I turn it over to Edgar, just a couple of procedural issues. So all participants are muted by default. So Edgar, if you are fine with this, we can take questions in the middle of the colloquium as well. So if any of you have a question, please either use the raise hand feature on Zoom, or you can use the chat box to type out your question. If you have raised hand, then one of the hosts will unmute you so that you can ask the question yourself. Or if you have typed it into the chat box, then we'll just read it out so that Edgar can address the question. And then at the end of the colloquium, we can, of course, have a more open discussion within the constraints of whatever time remains. Right. So Edgar, please take it away.
Thank you, Mithun. I'm really pleased to be here. This is an excellent opportunity and initiative, joint initiative between us and IIT Bombay. It's really exciting to be the first speaker. I want to say hi also to all the crowd, 63 participants at the moment. I see many people from India, so I hope this talk is interesting for you. So I'll share screen, and you tell me if you see everything as it should. Okay, please let me know if you see my slides and also my mouse. You see?
I can see the, yes, we can see it. Yeah.
You see the mouse. Okay, perfect. So, so thanks again for the invitation and for you to be there. Today, as Mithun introduced, I'll discuss on applications of the field of stochastic thermodynamics, which is an emerging field in statistical mechanics, on to understand the active fluctuations in the ear of the bullfrog. So this might sound to you a bit two distant fields, but I'll try to convince you that they are not so distant from each other, and that we can apply universal principles on thermodynamics to the fluctuations in the ear of this type of bullfrog that I will introduce to you in a few minutes. So this is a bit of an introduction on living matter and on how life works. So we know that there are many physical or biophysical phenomena that are out of equilibrium. So you see, for example, on the top right, microscope image of a cell division or a molecular motor on the bottom right, or this was the video of a flickering of a red blood cell, and this one was of a chlamydomonas swimming in a fluid. All these biological processes have some common grounds in terms of physics, and mostly they all have to fulfill some thermodynamic rules. In particular, these are all non-equilibrium processes, which are characterized by, okay, nowadays people call these active fluctuations, but we can just, as physicists, call these are non-equilibrium because they dissipate heat to the environment in their motion. So they consume energy that comes from mainly hydrolysis of ATP, and they produce entropy. They create disorder in the environment and they dissipate heat during their motion. So there are some universal principles that in principle should apply also to living matter. So over the last years, as Mitun was introducing before, there has been unprecedented advances, experimental advances for humans to interact with living matter and to get to know, let's say, the molecular fluctuations of a red blood cell or a DNA hairpin or an oocyte. And this has raised, so the fact that we can now do very precise experiments has raised the question of what can we do with the data from these experiments in terms of understanding, for instance, the thermodynamics of these fluctuations. So these are some questions that different communities like biophysicists or statistical people in statistical mechanics or even biologists are now trying to answer. For instance, if we can distinguish active and passive fluctuations. So you look at that, you put a probe on a biological system, this probe fluctuates, and you would like to know whether this probe is performing active or passive fluctuations. I will talk a lot about this concept, but this is really a hot topic right now in statistical mechanics. Also, if we can infer from the type of fluctuations that we see, how much heat is being dissipated by a living system, or if we can quantify the arrow of time. So if we can say how much irreversible is a biological process, and further theoretical insights that we can think about. So these are many open questions, and I'll try to discuss some of them by focusing on this animal, which is the bullfrog Rana catesbiana. So this is a very scholars American bullfrog, and I'll discuss fluctuations that take place in the inner ear on these cells that have some protrusions. I will give more details later, which I'll call the ear head bundles, that displays in a fluid, because in the inner ear we have a fluid called the endolymph. So when the sound is transduced, these fibers are moved left to right, and this dissipates heat. So we're trying to understand or to guess or to infer how much heat is dissipated during the hearing process of the bullfrog by looking at experimental data extracted from this process. This is somehow one of the key goals of my talk and of my research over the last years, and I'll try to give you some insight about this. But before, as I don't know which is your background, it's the first of this series of colloquiums, I will introduce a little bit of the anatomy of the ear of the bullfrog. So first of all, what is the ear and what is hearing? So this is a sketch here okay, this is a human ear. It's not a bullfrog. Do not have ears like this. They have a sacculus, but somehow the working principle is similar than in the bullfrog. So the ear is like a microphone. So in a microphone, you talk to the microphone, and so you give a sound, which is a pressure wave that enters the ear. And in the microphone, as you know, the sound is transformed into an electrical signal that goes to the computer, for instance. But we have already this type of microphones in our bodies, because the ear is doing this. The sound enters the auditory canal, it's a pressure wave that hits the eardrum. this makes that these bones that are connected, the eardrum to the inner ear, also move and vibrate. And this ultimately generates a traveling fluid wave here in the cochlea that stimulates some cells that then generate electrical signals. So in the end, this is a very, very complicated process, but the key point in the sound transduction happens here in the cochlea, which if we look at the cochlea, it has this type of spiral shape in which it's arranged into cells that have this structure and that are sensitive to different stimuli. So this is very particular because it looks like a piano. If you have played piano, you know that there are the tones of high frequency on one side and of low frequency in the other. In the cochlea, we have a structure like this already built in, and this happens in our ear. So when you put a sound that is a very high frequency, you will only stimulate these cells. And when you hear a sound that is sensitive to low frequency stimuli, you stimulate this part of the cochlea. This is really fascinating. And okay, this is an image of a piece of tissue of the cochlea. And you see that these are arranged in this type of structures that look like, I mean, modernist buildings, very strange. And they have fascinated the attention of many physicists. And we are still understanding their non-equilibrium properties. So I'll focus in one of these. So this is the epithelium of the bullfrog sacculus. So this is extracted after sacrificing a bullfrog. You extract the tissue and you can do electron microscope and see these structures. So you can take this structure out and you discover that this is part of a cell, which is called the ear hair cell. You can see this image here. These cells have a very big cell body. Here is the nucleus. And in the membrane, they have these particular structures called the hair bundle, which is a series of filaments that are arranged in this way, which have a size of around 7 microns. So in this talk, I'll focus on how these hair bundles behave and how the fluctuations are responsible for sound transduction. And I'll mainly try to discuss which are the thermodynamic properties of the oscillations of these hair bundles. Okay, so from now, when I say hair bundle, I will be talking about one of these. As I said, they can have different sizes. This is a bit of an average, and there are different. Some of them are sensitive to low frequency stimuli, and others to high frequency stimuli. So there is really, in the tissue of the ear of the bullfrog, you can get many of these sensitive to different frequencies. So how sound transduction happens? So in the end, the traveling wave in the cochlea bends all these structures. It makes that all these are called stereocilia, these long filaments. They are bent, and this creates that some other filaments that are called tip links, which connect each of these stereocilia, are stretched. And when they stretch, they generate the opening of an ion channel. So here, this is a microscope image of the ion channels that are present in the structure. So when the ion channel opens, they allow the passage of calcium and potassium ions, which then are the responsible of the electrical current that goes to the auditory nerve. So this is a bit
Edgar, there is one question. I'll just read it out from Anirban. Yes. So he's asking, how is this conical structure that you showed that is connected to the spiral that you showed before?
Yes, very good question. So if you have this spiral structure and okay, this has a basilar membrane. So if you take a piece of this tissue, and so if you, I don't know how to say, but maybe I can do an image. So if you take a small region here and you chop it, then you see this one. This is a magnification of a small region here. Okay, so this is like a very thin film in which there are all these cells arranged in this vertical arrangement.
Thanks. Yeah, thanks. Yeah.
Okay. All right. Okay. So I was discussing that this is the way hair cells transduce sound into an electrical signal, and of course this is not this is an active process because the ion channels are connected also to molecular motors that are responsible of adaptation. So adaptation is the phenomenon in which when you hear a very, very strong noise, your ear is able to adapt to very strong stimuli. And this is done thanks to the action of these motors that are moving also the channels, and therefore they are responsible of changing the length dynamically of these techniques. So here there is ATP consumption. This is an active process, and still we are trying to understand how much energy is dissipated in this phenomenon. But this is just to tell you a bit of the biology of this process. Okay, as a physicist, there are many fascinating features for this that have been discovered for the ear hair bundles. One is amplification. So they are able. So this is an experiment where they put one of these hair bundles in a plate, and they move the base of the plate. And this is the motion of the base, and they see that the tip of the cell moves with higher amplitude. So they can amplify sound or stimuli. Another important feature is frequency selectivity. So different animals, we are sensitive to stimuli of in a range of frequencies. So for example, lizards can hear sounds that we cannot. Nonlinear response is another important
Edgar,
property.
excuse me, there is another question.
Yes.
So Dibendu has asked, how do the hair bundles in different regions sensitive to different frequencies differ?
Okay, this is what I'm trying to explain now. So you can see this is experimental. It's not a model, and they could experimental techniques measure the sensitivity of this of the entire ear to different frequencies. It is still an open question how this works. So it's what we can do is to extract the cells, see how they look, and then try to guess why. So how can you make something, say, sensitive to one frequency and not sensitive to another? This is a big question. It's still it's an open question in the field. So we know it happens. We know because you can take out from the frog cells from different parts of the tissue and see that they oscillate at different frequencies. Okay, so this is just, let's say, an empirical observation as for now. And still, we're trying to understand why. In some parts, the ear is sensitive to high frequencies, and why in other parts to low frequencies. Okay.
Thank you. Yeah.
Welcome. All right. So another important phenomenon is that they have nonlinear response. So when you listen to the sound of a needle falling close to you, this is a given amplitude in decibels, whereas when you go on the streets and there is construction works, you will hear a sound that has 10 to the 4 times the amplitude of the fall of a needle. But your ear doesn't respond in a linear way. So if your ear would respond 10 to the 4 times stronger to the sound of a construction work, you will probably die. So you need a system that is able to respond to different amplitudes in a nonlinear way. And this is actually have been shown in many experiments. And finally, the one that I will talk most is something called spontaneous oscillations. It means that the ear doesn't need sound to oscillate. So if you look at these hair bundles and you put them in a quiet environment, they still vibrate and develop this type of spontaneous motion. So if you put yourself in a quiet room with a very, very precise microphone, you will hear sound coming from your ear. And this is due to the fact that there are molecular motors that are consuming energy, and they generate this type of active motion. So this is a very fascinating phenomenon, I believe. We are trying to understand how this works and how is this possible from a physical point of view.
So one more thing, Edgar, if I may. So there is another question that are the hair bundle frequency sensitivities related to the sizes of the bundles?
It's not clear. It's not clear.
Okay.
I'm not 100% sure. So I think it's not clear yet. It's not clear yet, because you can have also different hair bundles with, so here I show you, for instance, one case which have maybe 50 of these stereocilia, but there are others which have 30. So there's not a one-to-one relation between number of stereocilia or height and frequency, as far as I know. But I invite you to take a look to recent biology papers, because maybe this is better known right now. It's an open area of research.
Oh, thanks. And about the spontaneous oscillations, is there like some characteristic frequency that depends on the motor?
Yes. This is clear. So if you see here in the frog, you will see that there are some characteristic frequencies in the fluctuations, and this is what I'm going to show you in 3 or 4 slides. So you will see. So this time series here is not like a particle in a double well with exponential waiting times. It's not like that. And this is a key insight from the Hell bundle. You'll see. So it's an oscillation with a given frequency plus noise. You'll see. Okay.
Thank you.
Okay, I will talk about this in a few minutes. But yes, so physicists have tried to come out with models that describe this type of spontaneous oscillations. And at the beginning of the Hell bundle studies, there were many proposals were about the fact that the ear may be near a critical point. Yet there was this very influential paper that found that there was, you see, a critique of the critical cochlea. So it was saying a Hopf bifurcation is better than none. So here they propose dynamical systems near a Hopf bifurcation as paradigmatic models for these oscillations. You can find some information about this in the great book by Strogatz on dynamical systems, and think, for example, of a complex number, and here I'm plotting the complex number, the complex plane, which its radius and angle follow these equations, which are differential equations that are nonlinear. And you can have, when changing this parameter mu, you can change your dynamics in a way of Hopf bifurcation, which means that you go from a situation where there is one stable point to a situation in which this stable point becomes unstable, and it opens up, and then you have a stable orbit in which you perform oscillations. So, okay, this is no connection. I'm not talking about any biological parameter here, but this is just a physical system that can have these spontaneous oscillations. So this Hopf bifurcation can be of different types. I won't discuss much now, but I invite you to take a look to the book of Strogatz. But the main point is that this idea is very useful to discuss the possibility of having a model that has monostable solutions in which you have one stable point here from where you may escape because of fluctuations, or oscillatory solutions in which you have the system oscillating continuously. Of course, this was only the beginnings in which there were these models which have no noise. So one cannot have this type of oscillations with noise with these models. One has to add thermal noise, for instance. But yet, with this type of modeling, one can get very, very many of the key features of the Hell bundle, like very sharp power spectra, nonlinear response, etc. So it was a very important development in the field of Hell bundle modeling. Yet, however, it has been 40 years of modeling. So 40s, 2010s, and 20s, people have been developing different models, starting from dynamical systems close to Hopf bifurcation. In the 2000s, there were developments of models which are two-dimensional or three-dimensional Langevin non-isothermal models. It is not isothermal because we think of one variable is the top of the cell, another variable is where are the motors, and the motors are active, so their fluctuations are not in a thermal bath, they are in an effective temperature somehow. Then the next decade, there were many multidimensional Langevin models with nonlinear forces. So this is important that nonlinearities are key to reproduce most of the experimental data. And in the last decade, okay, there are many developments. For example, this is a very nice idea of a hidden van der Pol oscillator, or we just propose another model which is linear, but which has no Markovian noise, and we can solve. I will talk about this at the end of the talk. So this is one of the most important models in this. One of these, I think this was in the 2010s or so, which has is described by three nonlinear couple stochastic differential equations. As I said, this is the position of the top of the bundle, and this is the position of the motors. This one is the concentration of calcium in the cell. I won't enter into details here, but just to tell you that this model has been fitted to experiments. You see here, this is for the question of Mithun, the power spectrum density of the spontaneous oscillation has a peak. So they have a characteristic frequency, and this model can fit the experiment very well. Moreover, for different parameter choices, you can build this type of phase diagram in which you can have bistable oscillations, monostable, and also this oscillatory regime I was talking about. And an important question is, where is the experimental data in this diagram? So the experimental data from this paper is shown to be right in this point, so close to a Hopf bifurcation. So here we go from monostable to oscillatory through a Hopf bifurcation. So it has been really the, say, the bread and butter of the community to establish that the ear of the bullfrog is close to a Hopf bifurcation. So this is something, let's say, well accepted in the community because of these experimental advances. Okay, so these models, they are not only like nice to fit data, but they are also good to do predictions. And this is a very fascinating work where they coupled a real hair bundle to a computer. So they had a computer that was performing the simulations of this model here. So the computer is integrating Langevin equations and responding to the motion of the cell, the real motion, experimental motion of the cell. And what they observed is that, okay, first, they can have synchronization between these cyber clones or virtual cells and the real cell. And also that the response of the cell in this case is the same as if you have it in a real tissue. So this is a very, very strong way of validating a model. And it's really a crazy experiment if you think about. But then later on, it was shown that the head bundles not only oscillate spontaneously, but when they are close to each other, you can put probes on top of them and see that they, after some time, they can synchronize. So if you put them in a medium where they are connected but through their base, they, okay, there is a raised hand, they can synchronize among each other. Yeah, I think there is a question, so I'm happy to.
Yeah, I'll just unmute.
Please.
Hello. Hi.
Hi.
Hello.
Hi, hello.
Yeah, I had a question. If you can go back to the PSD diagram of the previous page. So which parameters does that correspond to? And just a quick question, what are S and F max again? I think I missed it.
Yeah, sure. Very good question. So this model has around 20 parameters. So I invite you to go to the paper and see the parameters because these are mechanical parameters. For example, the stiffness of for example, this is the sterocilia pivot stiffness. So it's the stiffness of the sterocilia to move in the basilar membrane. This is the stiffness of the tip links. So there are many mechanical parameters which you can extract from the fit. They are available here. And these two parameters are somehow the ones that they cannot be measured in any other way. So there are different ways to estimate frictions, stiffnesses, diffusion coefficients, etc. But these two, they are like free parameters across the fits. So one is the maximum, this is called F max, is the maximum force done by the motors. So we assume that the motors are fluctuating and they exert forces. They appear here on the bundle. And this has like a sigmoidal shape, and it has a maximum value, which is called F max. So this is the maximum force exerted by a motor of the adaptation motors. And this S is a parameter that is called the calcium feedback strength, which tells you how strong is the feedback of the calcium concentration on the motors. So somehow this is a parameter that relates C to XA, and this is a parameter that relates XA to X. In a very briefly, okay? For details, I invite you to take a look at this reference. Does it reply your question?
Thanks. I think you can continue.
Okay. All right. So, okay, this is another fascinating phenomenon that we observed. And as from now, I will focus among all these features on the spontaneous oscillations. I'll try to understand what can one do with the experimental traces. Experimentally, one can put a fiber close to the Hertz bundle, as we show here, and move the deflection of a fiber as a function of time, which has this type of shape. This is a typical time series of 20 nanometer amplitude and frequency of typically, for example, here before I was showing a frequency of 8 Hertz. So in the bullfrog, we can extract and resolve oscillations of 8 Hertz in this case. So, of course, experimental life is quite hard. So I think most of you are theorists, so I will skip this. So I tell you there is a fiber that we can look on top of it, but this is taken from the PhD thesis of my colleague, and it's a very complicated device. So when you see one day time series, please take into account that this took a lot of work and calibrations. And I start with my questions for you. So what can T.H. Smith do about backtractions? And you may ask what is T.H. Smith. This is a bit of a joke, but I take it from this paper by Christian Miles, which says T.H. Smith is the statistical mechanician in the street. So someone like you may be a statistical mechanician and would like to know, if you get data from a biological experiment, what can you do from your street knowledge on statistical mechanics? So the first thing we may wonder is, you take an experimental time series and you would like to know if it's equilibrium or non-equilibrium. This work answers to this question by revealing that these fluctuations are active. So please take into account that this is 2000. So now there is a lot of work on active matter, but this active matter has really a long story. So just keep an eye on this paper. And what they do is they compare the spontaneous fluctuations of the bundle to the response. So they look also at the bundle when this probe is exerting a sinusoidal force. So we will get 2 time series, the spontaneous and the forced time series, and one can analyze them and try to see if with this 2 time series one can say that this is equilibrium or non-equilibrium. So to answer this question, we can use common knowledge in statistical mechanics, namely the green Kubo, the Kubo fluctuation-dissipation theorem. So as you know, you can take a colloid, for example, in a thermal bath, have a time series, and compute the autocorrelation function of the time series, which is defined like this, and from it obtain the Fourier transform, which is called the power spectrum, mainly. And on the other side, you can do a second experiment where you drive this colloid. So there is a trap, and you move the colloid sinusoidally, and look at the average motion of this colloid as a function of time, and build from this the linear response function. So you assume the force is small, so you put a small force, sinusoidal force, and you can extract this quantity, which is the response function, which is typically complex, so it has a real and an imaginary part. So Kubo found that if the bath is in equilibrium, these 2 quantities are related through this equation, which is called the fluctuation-dissipation theorem. This part is the fluctuation in the sense that you just look at the spontaneous fluctuations, and this is the dissipative part of the response. And you see that here the relation is through the temperature of the bath. So you must have an equilibrium thermal bath driving these fluctuations. So let's do what statistical mechanician can do, is to test if this theorem works. So you get a time series, you see the stationary density is bimodal, and you can compute the autocorrelation function, which has this type of oscillations, and from it the power spectrum. So what I showed you before is just the Fourier transform of this function. On the other hand, you can force the Hertz bundle with a sinusoidal force of different frequencies, and see that the response function to this force has a very particular behavior, because it has a peak. The real part has a peak, and the imaginary part changes sign in a given frequency. And this is exactly at the frequency of the spontaneous oscillations, 8 Hertz. So now you take this one and the power spectrum, you compare them, and you show that if you do these 2 experiments on a passive cell, so post-mortem, the fluctuation-dissipation theorem holds for any frequency. So you do the experiment at different frequencies, and you get the fluctuation-dissipation is fulfilled. On the other hand, when you do this to an active cell, you see fluctuation dissipation is broken, and this is a signature of the fact that these fluctuations are non-equilibrium. It's a very, very strong proof. However, you may want to go deeply in this question. So it's not only a binary question, are you in equilibrium or not? Well, of course, we guess it must be out of equilibrium because it's an active process. But you would like to know how far from equilibrium. You'd like to quantify the entropy production, for example, in the motion of the hair bundle. And for this, we published a paper last year, but it took us a lot of time. So I started this as a PhD student. So it's 9 years of research. It's a long time. So I'll try to give you a fresh update on this. So first of all, there is this fact that currents are a footprint of irreversibility. So if you think of a simple model like a Markov process with 3 states, if you are in equilibrium, you will have no current, so no net current between any 2 states. And this will imply that the entropy production, which is usually formulated as the sum of suitable forces times currents, is 0 in an equilibrium process. Let's say when you have a Markov chain with all the rates fulfilling detailed balance. However, when you have non-equilibrium stationary states, it means that you are biasing the rate. For example, here you go more clockwise than counterclockwise. You have currents between all the states, and this is a signature of heat dissipation, so or entropy production, which typically takes this form. So in the last years in the field of stochastic thermodynamics, there was a big development on how to compute the entropy production the entropy production rate of a process, for instance, a Langevin dynamics in 2 dimensions, in terms of stochastic trajectories produced by this process. And here I cite a result that is valid for all non-equilibrium process in the stationary state, in which we do the following. First, we look at the trajectory, a single trajectory in a phase space. For example, here is 2 variables, so you have this sequence of states. And you look also at the time reverse trajectory. And of course, if this is an equilibrium process, you will see more time the blue than the red. Sorry, if it's equilibrium, you will see the same number of times blue than red. But if it's non-equilibrium, there will be drifts. So you will see more times this trajectory than this one. It turns out, and we can show in stochastic thermodynamics, that computing the Kullback-Leibler divergence, which is, let's say, this is a measure of how different is the probability to see a trajectory with respect to the probability to see the time reversal trajectory. If we compute this quantity, this is related to the entropy production, and in fact, is a lower bound always to the entropy production. So whatever coarse graining you do, if you build trajectories and you do probabilities of trajectories and time reversals, this quantity will give you a lower bound to the real entropy production, which is a physical quantity. So on this left-hand side, we have a quantity that it will depend on the specific model. So some models are isothermals, and this is just the heat over the temperature. Other models have 2 thermal baths, and this takes a different expression. So this is the physics. Whereas this on the right is just a statistical quantity. So it's just computing probabilities of trajectories and reversals. So of course, your estimates to the entropy production will depend strongly on how many variables you see. So here we can see with 2 variables, we can see currents and compute the reversibility with 2 variables, which is close to the entropy production in some examples. Whereas if you don't see the 2 variables, for example, you see only x1, you have like something oscillating, but you cannot see current. So it's very complicated with 1 variable to see entropy production. So these bounds that we establish are not always informative. So if you reduce a lot the amount of information, you will get very, very weak bounds to the entropy production. And this is the case of the hair bundles. In the hair bundle, in the experiment, we only get 1 variable. So it is really challenging if we want to estimate entropy production from something that doesn't have a current. So you go the same number of times left than right. You are oscillating. So we try to do this to estimate entropy production from one variable in 182 recordings of, so these are 182 bullfrogs. Each recording has 30 seconds.
Edgar, excuse me, there is a question. So the question by Rajarshi is like, what happens if the dynamics in underdamped, if the dynamics is underdamped, where velocity is also a slow variable? Will your formalism for entropy production and bonds hold?
Yes, so here I'm assuming overdamp. All the variables are overdamp, but you can extend this to underdamp. The only thing that you have to be very careful is that in underdamp, they are not only x's, they are also v's. So when you time reverse the trajectory, you have to change the sign of the velocities. So you can do it in underdamp. You can do it in underdamp, but instead of having only x's, you will have also x. It will be x1 and v1, x2 and v2, etc. And here there will be minus v's. So one can do it also with velocities. Okay. Yet experimentally, it's very difficult to measure the real velocity. But one can extend this. I can give you reference if you are interested about this.
Thank you. Yeah.
Okay. All right. So this is our challenge. We'd like from this time series to estimate the underlying entropy production. So I repeat, we need to compute the probability for trajectories and the probability of their time reversals. And there has been many developments with, for example, using the Grasberger-Procaccia algorithm, which I call Coulomb tasks, because they basically try to compute, so they discretize the time series and they try to compute probabilities of very long sequences. And you see, you have to do t going to infinity. So that's quite complicated. So we introduced a trick, which was the following. We take the time series and the time reversals, and we apply to them a whitening filter. So what we are doing is mapping. So we pass this by a machine, gives you a new time series. And now we pass the reversal time series to the same machine and gives you another time series. I'm skipping details, but this is called actually in machine learning, they use it a lot. It's called whitening transformations, in which you do a one-to-one transformation between the original trajectories and the new trajectories, such that the new trajectories are IID. So the original time series have correlations, and this one are IID. And yet, using the same transformation, you can see that the distribution of this IID process are different when there are irreversibilities in the original time series. So this map is very efficient because you map the original, so the original autocorrelation function is like this, and the autocorrelation function of the whitening time series is 0. So you can do this very, very easily with a computer. And the main advantage is that this simplifies a lot the calculation of entropy production. So when you are in an IID process, you don't need to compute anymore the probabilities of the entire sequence, but the one-time probability density. Okay, it's a major simplification. So you just do the transformation, you have two new time series, and you compute the histograms. That's it. You don't need anymore to look at trajectory probabilities. Okay, I'm skipping many details, but this is the trick. I'm just trying to give you a first smell on how this works. And for more details, I encourage you to look at the paper. Or I can also explain you later more. So if we apply this technique of whitening and we compute irreversibility by this difference of histograms, we can apply this to experimental time series and to also, let's say, a negative control, which is a particle jumping in a double well. When a particle jumps in a double well, if you assume this is equilibrium, you will have also these jumps, yet you know it's equilibrium. So when we apply this measure as a function of the number of data that we use, you see that this case gives you 0 irreversibility within the error, whereas the experimental cases, they give you positive irreversibility. So we can distinguish between these ones, which are hard to say they are irreversible, from a purely equilibrium case. So this seems to work. Moreover, we can apply this technique to different conditions. For example, these are cells that are active, so they are consuming ATP, and you see that this irreversibility is larger than when we take the cells and we drag them. So we put a drag that doesn't allow the channels to open and close, so they are somehow passive, and they have less irreversibility. And this irreversibility is of the passive is comparable to the one of the experimental noise. So we can classify different cells in between active and passive very efficiently. Yet, something I didn't say is that how this sigma 1, so this irreversibility in the Whitening time series, compares to entropy production. This is an open question, and what we try to do is to use models, because we don't know the second variable. In reality, typically we have 2D oscillators. We don't know the second variable, the position of the motors, but we can use simulations of the model, like I show here, to compare the irreversibility in X1 to the full entropy production in this phase space. And the main result is that in this Fmax S diagram, in the oscillatory regime, we detect, in the oscillatory regime, we detect irreversibility. This is the place where there is entropy production, but yet the order of magnitude is very low. So we say 10 to the 1 here, whereas entropy production is 10 to the 4. So we are still far with sigma 1 to detect entropy production compared to the case when we have two variables where we see these things very well. We almost collapse the real entropy production. So this is a bit of a pity from this approach. So we try to get one step further, and the step further is if you are able to infer the second variable. So of course, this you could do by imposing this model using machine learning and getting the time series X2. This would be a very beautiful approach. But we try to do an approach that doesn't require machine learning, that is simplifying this model and have a second variable that is easier to infer. And this is what we did in this last paper, introducing this non-Markovian model. Okay, I think I'm a bit tight in time, but I'll try to give you a brief idea. So this is an effective model in which we say that we have a particle in one potential, and the potential is switched to another potential. You go from blue to red, and these are two harmonic potentials that have different centers. So you are switching the harmonic potentials at random times. Okay, so the particle is trying to follow these switches, and you see here the dashed line is the C process, which is jumping between two states, and the blue is X process, which is trying to adapt to these jumps. The main point is that these switches happen with a time distribution that is arbitrary. We set it arbitrary. So it therefore breaks little balance between the jumps. So it's a non-equilibrium process. The nice thing of the approach that we introduced in this preprint is that we select, so we put the two distributions, the top and in the bottom state of the C process here, of the center. We set it as arbitrary. So there are two waiting time distributions that are arbitrary. So if you put them exponential, you will have a Markovian process whose power spectrum looks like this, like a Lorentzian. Whereas if you put them non-exponential, like they have a peak, you will have a non-Markovian process, very important, and the spectrum has a peak. So this looks more like what we see in the bullfrogs. Moreover, we can solve, okay, there are two big classes of models when the exponential times are exponential and when they are not exponential. When they are exponential, we can solve the problem analytically and see, for example, that the distribution of the tip of the bundle can be unimodal or bimodal. So we find that there is this type of phase diagram, which depends on two parameters. So this R is the average time between the jumps. So when you switch very, very fast, you cannot be bimodal. So you need to switch slow, but yet the C0 cannot be, the C0 has to be large enough so you can resolve the two peaks. So this we can derive analytically, and it resolves somehow all this part of the diagram. So we can take, we can describe this model monostable, unimodal, and bistable. This part yet is not taken into account in our model. So it's a model valid for all this region of the Hermann. So why I'm saying that we don't use exponential distributions is motivated by experiments. So we took experimental data and tried to fit the waiting time distributions to try to fit it to any function. We found a very good agreement between the experimental data and a gamma distribution, like the one I show here, which is a power times an exponential. this motivated us to introduce this model, but it also allowed us to do predictions on the data. So here I introduce you to Alfred, which is one of our bullfrogs from Jim Haspel's lab. And here you see the experimental time series and the stationary distribution. And what we do is we compute the autocorrelation function and we fit it to the analytical value. So for this case, even though the psi's are non-exponential, so the dynamics of X is non-Markovian, we can still, even though it's non-Markovian, find analytically the expression for the autocorrelation function. So somehow we take Alfred, it's here, we look at the time series, we build the autocorrelation function, and we fit it to our formulas. And from here, we extract the parameters. So somehow it's an advantage with respect to previous models, which were not analytically solvable. So we can now do predictions of the Hermann. This is Alfred. Okay, this is a power spectrum, but we have also Manfred, in which you see the agreement between experiment and the theoretical fits are perfect. And this is Lothar, and Lothar also is very well fitted by this minimal model. And then, okay, this is the last thing I will tell you is that not only we can fit the data to a model, but we can do predictions for thermodynamics, because now this is a very simple model in which you introduce energy whenever there is a jump. So when the particle jumps up, you are exerting work on the system, and whereas when the particle jumps down, you are extracting work. So we are computing somehow the energy changes of the system whenever these switches happen. Okay, this is the theory, but what we can compute is how this changes in time on average. So if there is a power dissipation in the ear, we get this nice expression, which depends on the parameters and also on the Laplace transforms of the waiting times distributions, which we say is gamma. So it's a very simple formula evaluated at the this is our characteristic frequency. This is the stiffness of the trap divided by the friction. So this is a very nice formula that is generic for any waiting time distribution. And now what we do is we take the data of Alfred, Manfred, and Lothar, we extract the parameters, we plug in here in the power, and we get an estimate of the power dissipated in these oscillations, which is much bigger than what we are getting with the irreversibility measure. The irreversibility measure was like 0.01 here. You get around 100 kVts per cycle. Sorry, in the irreversibility measure, we were getting 1 kVt per cycle. Now we get 10 kVts per cycle. So it means that for every oscillation cycle, you have to burn an energy of order 100 kVts. So it is not a fluctuation. If it's a fluctuation equilibrium, it will be 1 kVt. So this is really a clear signature of activity and of dissipation in the system. And this coincides, is the same order of magnitude as the full entropy production that I reported before with the more complicated model. So this is a simplification, and it's a very robust tool that you can fit to many things. And indeed, with this, I will give the last technical slide, which is there is a lot of interest in the last few years, in 2019, on revealing irreversibility in waiting times. It's not only us, but this very beautiful paper by Martinez, Bisker, Parrondo, Horowitz, Nature Communications, which also pinpoints that if you have asymmetric waiting times, you can detect irreversibility. There is also this really good paper in Nature Physics where they look at a cell crawling in this two-chamber compartment and look at the waiting times in each state, and it's also like non-exponential, like the one we use. So one could use our technique to estimate the dissipation in this model. There is also this very crazy application of irreversibility. So entropy production estimate from waiting times in cows. So you can look at a cow, how long it stays sleeping and awake. And they estimated in this PRL paper entropy production from cows. and finally, okay, we will also have a contribution on more theoretical coming next week in archive about also waiting times, which I hope that you can read soon. So with this, I will conclude. I have three main conclusions. So the first is that stochastic thermodynamics has been applied through decades to very, let's say, a reduced set of experiments like colloids, molecular motors, linear hairpins, electrons. But now I think the best is coming, because still there are few applications on real biological process, and this is one of them. Like we are highlighting this, and this is only one of the many possible directions. Second is, okay, for bullfrogs, we estimate a dissipation of around 10 ATP per cycle. So I said around 100 kVts per cycle, and this is around 10 ATP. So maybe we are starting to infer the number of ATP molecules necessary to drive the system. And this is an important result, because the molecular motors is an hypothesis. In the end, they know there's myosin, but nobody has seen the molecular motors in the microscope moving, because these experiments, they do cryo-EM, so they kill the cell to see the structure. And then, okay, the truth is yet out there. I mean, there are still many things to do, like look at the power and efficiency of sound transduction. So we are using spontaneous oscillations. We are not looking at the transduction yet. The phonetic aspects, it's another direction in stochastic thermodynamics. Ideas from optimal control, etc. So there's much things to do. As you see, there are many open questions, yet this is a system that is known since 40 years. Okay, I would like to finish with acknowledgments to all my collaborators in these adventures on the bullfrog, and also my group at ICTP right now, the people who's working with me directly. Also the QLS section, in which I'm working since 4 years, and it gives a great environment. I want to highlight two people here in this list, Roman and Gennaro. Roman has been postdoc here 2 years and has done most of his work on the last part, and also Gennaro did the PhD here in CISA, and now just moved to MPI Göttingen for a postdoc, and the last work is mainly thanks to him. I would like to thank you for your attention. This is the place we work, and this is the website from our institute, and I leave you with this beautiful Batrachian for the end of my talk. Thank you.
Thank you, Edgar. That was a really nice talk. So I think we can take questions. We have some time. So okay, so let's see. Please, if anyone has questions, please raise hands, and we'll then unmute you and you can ask the question. So I think Ishant has a question. So Ishant, please.
Hello.
Please, please.
Hi. Thank you for the informative talk, Professor Roldan. I just have two questions I would like to ask. First one is, in the end, you mentioned this switching of harmonic potentials when you simulated. So I want to ask, maybe I misunderstood. So is something similar happening in the hair bundles inside the bullfrog? Is the equilibrium of the hair bundles shifting from one to the other? Is that why the model is predicting so well?
Yeah, indeed. So if I show you the time series, okay, there's a big collection of time series, so I don't think I can give you all of them, but if you look at the time series, you see that it looks like that there are two stable points in which you are jumping between each other, but there is a relaxation, so this doesn't happen instantaneously.
Okay.
So if you zoom in this time series, you will see a relaxation towards an equilibrium point, more or less. But it depends a lot on the time series. So some of the time series look like this, and you can describe this model. But others look like sharks, so they are extremely reversible. They do.
Okay.
I don't know if I can draw here, but some time series look like this. Okay, so there's a big phenomenology, and we can fit a subset of all the oscillations, mainly the ones that look bistable, like the ones I show here.
Okay. Thank you for this answer. I have just one small question, another question, which is when you mention the spontaneous sound that one hears in a quiet room, are you talking about tinnitus? That sound that when you are you hear an explosion or something. That sound.
No. So I can tell you a very fascinating case. Okay, of course. What I'm showing, the last data is just a single cell. So this is a sound that you will not hear unless you have an incredible amplifier. But I can tell you a case that I knew in Germany of a patient who had some strange mutation in the ear, and it was a family, and this family at night they were realizing that the daughter was somehow producing sound, and they didn't know what was going on. So it seems like some humans, they produce sound from the ears. It's like a spontaneous production of sound that can be measured with a microphone. So it's really at the physiological level. So we are continuously producing sound, even though if you put yourself in a quiet room, you'll be still producing sound because the motors are alive. They are consuming ATP and moving these structures. So it's I don't know if this answers your question, but this is what I meant somehow.
Okay. Thank you so much for your answers.
You're welcome.
Srilaja, you can go ahead, please.
You can go ahead with the question.
Yeah. So I had a couple of questions actually. So one of the questions was in the theoretical models that you are building, how do you account for attenuation of sound? So in biological systems, the sound stops, therefore you stop hearing it. But does stereocilia have a period of recalcitrance? Because it is a mechanical perturbation, it requires some time to reset back to state zero. So in the theory aspects, how is this accounted for?
This is not yet accounted because this model is for a single cell. It's not for let's say, a collection of cells. So you would like to see like the traveling wave and the cochlea passing through a collection of cells. This is not accounted by our model because we are at the level of a single cell. But there are very interesting recent modeling from, I think the Bolshevik lab is doing this in UCLA, in which they model a collection of cells coupled to each other, in which a stimuli is passing, and it has a spatial structure as well, it's space-time. So one would need to extend the model to many degrees of freedom, but in principle, one should be able to do it.
So in theory, if you multiplexed cells, because stereocilia, the cilia are arranged in a particular pattern, and that pattern is evolutionarily.
Okay. So sorry, there's another paper you can take a look, which is I will write in the chat. Take a look. Okay, I'm trying to send. I think it's a paper by Baumgartner, Johannes Baumgartner et al. I forgot the year.
Okay.
It's in Nature, not Nature Physics, Nature. This is a very good model which is a mechanical model, let's say engineer type of the stereocilia with all the elements and so on. These type of studies were very popular also, and they give an idea of this. But it's not, let's say, I'm not an engineer. I look at very tiny things, which is a single cell, and I'm doing a very effective model in which I'm ignoring really the atomic resolution. But if you look at this paper, you will see some work in this direction.
Okay. My other question, I got the chat thing, I looked that up. My other question was, at a single cell level as well, when cilia are lost, when a collection of cilia are lost, would your model be able to tell what are the, let's say, the breaking points of that cell, which when hit, the cell stops responding?
Can you repeat the question?
So the model is based on the movement of one cell's cilia.
Yes, okay. One important point, and this is also the question of Arnab Pal. is what you look is at the motion of the tip of these cells. You see, this is this is the outer membrane of the cell and this protrusion. So you look at the motion of the tip of the hair bundle. It's a single cell. Yes. Okay. But your question is,
my question is. So generally, the sound wave is encountered by the front bundle or the back bundle, and it matters like where the sound hits. So in your model, is it possible to reverse the starting point of this wave to see how the wave propagation would be perturbed? Would the wave propagation be perturbed at all? And if so, how?
You will need a model with many of these x's, and then you have to introduce another variable and also external forces that have the properties that you look. So from the model that I just showed you before, typically these models, okay, they look like this. Okay, this is for one cell, so you will have to extend the model for many cells, and then there is an external force that is coming to the cell. So you need to add these forces and tune it in the way you would like to look. Moreover, these x's will be different in different positions, so the model will become very complicated. But it's a good direction, I think.
So this is the equation on the basis of which I was asking you the question, because you have fixed the position, right? You fixed the position where your wave begins.
Yeah, sure.
Maybe biologically also that is true, that's the wave always begins in a particular position on the cell, but may not.
Okay, this is constrained to, you take the ear of the bullfrog, you do a section, you take one of these guys, boom, you put it in a plate, and you look at the motion. This is what we are describing.
Yeah.
Not an ensemble, because doing an experiment with an ensemble, it's very challenging, and you cannot have this resolution for an ensemble. You will have to do video microscopy, and this is less accurate. So yet it's a bit science fiction, but one day it will be reality. So it's good to do for the future, actually.
All right, thank you.
Welcome.
Can I ask?
Rajarshi,
So this is Rajarshi Edgar. Now, Edgar, I have a very general question. It's more about the physics part of it. This is regarding entropy production. Now, how the way one defines this entropy production, how unique the definition is? Because you can have a model 1, model 2, model 3. You can break your time reversal symmetry by introducing noise, or you have two harmonic wells switching between them. Numerous way you can do. And for each model, you can define some sort of entropy production. And then I'm just curious what is the status of this entropy production business in active matter right now? We wrote some papers a few years back, but I'm not much sure.
Okay, you're asking many questions. It's
a general question I have.
So typically, in stochastic thermodynamics, we have a system with states, and we say that the rate for A to B to happen divided by the rate of B to A is related to the environmental entropy change in this process. This is an assumption that we use always in stochastic thermodynamics. So for instance, this would be in an isothermal bath, this would be the heat over the temperature. So you need an assumption that the transition rates are related to physics, to heat. So we have this type of assumptions, and then if you have this type of assumption, you relate the environmental entropy to probabilities of trajectories given their initial state. Moreover, this is a very long story, but typically you describe the entropy production as the log race of the path probabilities, and then the point is, where does the physics come from? So when you specialize this quantity to specific models, you start to see the physics. So if you have a Langevin model in an isothermal bath, you compute this quantity and you get the work minus the free energy. So this is if it's isothermal. If it's not isothermal, you compute this quantity and it's the system entropy minus the heat fluxes in each of the baths, for example, in a Feynman ratchet. So it seems very obscure, but when you apply it to specific examples, the usual expression for entropy production specific to each model appear. this in the sum in active matter is an open question, so you can still compute this quantity, is the irreversibility, but typically you are ignoring non-equilibrium degrees of freedom, so this doesn't give you the hit, it gives you only a lower bound. So one has to be careful in active matter to call this entropy production. One has to be careful. So it's more the degree of irreversibility than entropy production.
Thank you. Thanks.
Welcome.
So the next question is by Arnav. Arnav, please.
Yeah. Hi. Hey, hi, Edgar.
Hi,
this
hello.
was very interesting talk. Very much liked it. Yeah. So I was just curious about this physical. I mean, I understand in the model you went beyond like exponential waiting time to non-exponential waiting time. Just I was trying to understand, like also in experiment, you indeed see a non-exponential waiting time which you feed with a gamma distribution. But do you understand what is happening? Because you say that essentially there is a switch between two states also in the real experiment, right? The one which you are showing. So do we understand why actually there is a non-exponential waiting time in the system itself, in the real system?
Well, I don't know how deep is the question, or do you understand, but this is an empirical observation. So we never see exponential distributions in the bundle, never, because this is a system that has, so it's an oscillator which has a characteristic frequency. Exponential waiting times, I've never seen in the 182 cells that I look, never. So one way you can build this is if you think on a model which, okay, this is something we were thinking for a long time. So if you think the model of the hair bundle, let's say it has, okay, this is a very bad drawing, okay? You have this model in which, okay, now there are distinct links, let's say like this here and like this here. And this bundle at some point will move to the left. So it will, this is one equilibrium position, and the other one will be all these three coming here, no? Okay, so to switch, at some point you need to take actions on this channel. So you need to pass from closed channels to open channels. And you could do a very minimal model, which is each of these channels throws a random number or has a waiting time to be open. So each of these has a exponential waiting time to pass from open to close. And then you should have to wait until all of the channels are open. So you go here.
So collectively, it's a collection of exponential waiting times, essentially. Which
yes.
Saying.
So exactly. So if you combine a collection of exponential waiting times, you will get something like this. But this is really my hypothesis. So it's nobody has thought about this idea before. So it's it's a nice hypothesis, and actually this k here is related to the number of exponentials if you want to build it like this, and the k we get is around 10, which is close to the number of ion channels.
So it just reminds me of this Erlang distribution, which essentially has this, like collection of waiting time, exponential waiting times, and the number of steps essentially gives you this k there. So
yes, exactly. So this is our a bit of our hypothesis. We are trying to explore it. So it's a bit intuitive by now, but we're trying to verify this in more experiments.
Right. And also, just almost a similar question. So in both the cases, actually, you see the bimodal distribution, right? For the stationary case, right?
What do you mean? What do you mean by both cases?
So both in the exponential and non-exponential waiting times, right?
Yes.
So how do you actually differentiate between them? I mean, like in real experiment, let's say that. I mean, okay, I understand from the parameter perspective, you will probably feed something and get this.
Yes.
But just looking at the bimodal distribution, it is not possible for us to infer something about the waiting time, probably.
No,
right?
no. That's clear. That's clear, because the stationary distribution does not depend on the correlation. You can have different correlations and obtain the same distribution. So you need, the way we feed the data is we feed the distribution and the correlation. They are two things. They are two phases of the same problem, but they are independent of each other.
Nice.
Okay, so it's different. This has information of one time. This is information of 2 times correlation. And in the non-Markovian case, you have to go to beyond in reality. But as for now, this is what we can fit experimentally. Yes.
Okay. Thank you very much, Edgar.
Thanks, Arnav.
Again, it was a very nice talk.
It was good to see you here.
Same.
Okay, so I think there are no further questions, and I think we've overshot our time also by quite a bit. So again, I think from all of us, thanks, Edgar. It was a really, really nice talk. Thanks to all of the participants as well for attending and for participating through the discussion session. The next colloquium in the series will be on the 13th of April. So remember, again, it's the second Wednesday of every month. So the next one is on the 13th of April, and it'll be by Professor Roop Malik from IIT Bombay. So hope to see all of you there for that talk as well.
We can stop. Great.
Thank you.
Thank you. I think I was a bit long, but I hope people enjoyed it.
No, it was really, really, yeah.
Thank you. Let me know about the recording when you have a